Hello, I'm going to try and get you guys on track with the analysis for the population genetics and evolution lab that we did. So, just a few things to remind you of. We have these two equations for our Hardy-Weinberg calculations. So, P plus Q equals 1. Remember, we're talking about the frequency of an allele. Now, technically, we say like dominant is P and recessive is Q, but remember, there are times when alleles are codominant and there's not just a dominant or recessive. They both show or they blend. So it doesn't actually matter whether you say that this is going to be allele number one and allele number two, you're still going to get the same answer. P squared plus 2pq plus q squared equals 1. That's going to be the frequency of the genotypes. So now we have to actually know or at least estimate how many of those organisms of the total population are homozygous dominant, heterozygous, or homozygous recessive. All right, so when we did our lab, everybody had 4 gamete cards. And when we started, if you remember, everybody in the class was heterozygous. So this is just fictional data that I made up. I know every class had a different size and different results. So we're starting out with everyone heterozygous, and then we go through 5 different generations. And we're going to say in the end, these are our class results. Now, what we need to do in order to figure out P and Q, I think this is where some people are getting confused, are kind of what we've done before with estimates is if we didn't know homozygous and heterozygous because they all look dominant, we would try and estimate those frequencies based on Q squared, based on how frequent that recessive genotype shows up. So if that was the case, I would go 4 over 22. That would be my Q squared. I would square root that to get P and then do 2PQ and P squared to estimate these. However, we are not estimating here because we actually know how many heterozygous and homozygous dominant there were. And so if you did that estimate that I was just talking about, you probably wouldn't get these same numbers. Maybe they would actually estimate that this is 10 and this is 8. And so, where you can, it's much more accurate to count the alleles because then you actually know how many there are rather than just estimating based on Q squared and Q. So, in order to count these alleles, we're going to start at the beginning. So, we had 22 people, which means we have 44 total alleles in this class, and 22 big A's and 22 little a's. So, that's pretty easy. Then, when we look at the end, if I look at these results, when I want to count the big A's, I have to remember that if 6 people are big A, well, they each have 2 big A's. So that's actually going to be 12 big A's. Plus, there were 12 people that had only 1 big A. So I have to go 6 times 2 is 12, plus another 12 from the heterozygotes is 24 total big A's. If I look at the little a's, I've got 4. So times 2 is 8, plus another 12. So that's going to be 20. So now I started with 22 and 22, half and half. I ended up with 24 dominant and 20 recessive. What you'll have to do at the end of this lab, I think initially they say, like, do you think that this population has changed? And that's just kind of up to you. Do you think a 4 allele swing is a big enough change to say that evolution has happened or not? Well, when you get to the last page, you have to do a chi-square to see whether or not it actually is significant. So when we do these chi-squares, we've done these a few times. So we've decided, like, if we did a treatment group in photosynthesis, your null hypothesis is the plant growth between the treated and the untreated is not different, or there's no relationship between the independent and the dependent variable. When we got to genetics, we were testing how close we were to predicted genotype ratios. Now what we're doing is we're going to test for genetic population Hardy-Weinberg. So Hardy-Weinberg equilibrium tells us that the frequency of the alleles are unchanging over time. So that's our null hypothesis that we're going to test. If that is our null hypothesis, then our expected is going to be the starting allele numbers that we had at the beginning of the simulation. Because if we truly are having unchanging frequencies, then if you start with 22 of each, then you would expect to end with 22 of each. The observed is actually what we get at the end. So there I put in our ending 24 dominant and 20 recessive. Now I just do the subtraction, square, divide by expected, la, 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 and I got 0.36. Be careful because your expected is not always half and half. In our simulations that we did, we always started out half and half. But let's say a population started out with 80-20. Well, if it's unchanging, you would expect it to still end 80-20. So expected is not always half and half. It's just your starting population alleles. All right, so I got 0.36. When we look at our formula sheets, when we look at the chi-squared, remember degrees of freedom is categories minus 1. So I have 2 categories of alleles. And so my degrees of freedom is 1, which means my critical value is 3.84. We have 0.36 as our chi value, so we are well below the critical value. So we are going to fail to reject the null hypothesis. So the null hypothesis then says that things are not changing, and we can't really reject that because it looks like things really haven't changed significantly. There's one thing that we haven't done yet. Going back to like this situation, remember I said that you could use q squared and q to estimate p and p squared and 2pq. One thing that you actually can do is run a chi-square and see how close you are to the predicted Hardy-Weinberg frequencies. So in that case, you would run a chi-square where you actually have 3 categories. You'd have your big A, your big A, little a, and your little a, little a. Your expected numbers would be taking this q squared square root, take it times the total population, calculate 2pq times the total population, p squared times the total population, and run a chi-square and see how close you actually are to the predicted Hardy-Weinberg numbers. I don't know if we're going to do that at some point, but if they ever asked you to on an exam, that is a possibility. Otherwise, we look at the frequency of the alleles. But when we do the chi-square, we're going to compare total alleles. We don't want to say we expect 0.5, 0.5, and we got 0.55 and 0.45. We actually want to use the raw numbers of the alleles. So I hope that helped you guys. I'll be back in school tomorrow if you have questions. Good luck on your quiz, and we'll see ya.